Alka, it is my pleasure to present our work on AFconverge, which is an alignment-free method for comparative genomic analysis of convergent phenotypes. So a big goal we have in biology is understanding the relationship of genotype and phenotype. And sort of doing experiments, there's a few different ways we can do this from observational data alone. So one very common approach is to use population genetic data in the form of a GWAS study. And this has several advantages. So particularly, we can get very large sample sizes, have power to detect small effect sizes. It is fairly easy to correct for confounders, and therefore we can establish, in many cases, causality between genotype and phenotype. Of course, there's also some disadvantages to using population genetics. So we have limited genotype diversity, and we also have limited phenotype diversity. And so we can contrast that with what we can get when pursuing the same question using comparative genomics. So this approach, on the other hand, has many disadvantages. We have fundamentally limited sample sizes. There's only so many mammals that we have, and there's only even fewer of them that have a particular phenotype of interest. We have impossible to correct for confounders. So, for example, body mass is confounded with longevity and also confounded with marine habitat. But on the other hand, there are some advantages. So we have much larger genotype diversity, which, depending on how you look at it, could be an advantage or a disadvantage. But one thing that is definitely an advantage is that we have very large phenotype diversity. So just to illustrate the phenotype diversity, here's a person holding the world's smallest mammal. And here on the right, we have the world's largest mammal next to a boat full of people. And indeed, their body size is many orders of magnitude different, despite actually, these are mammals of relatively short evolutionary distance. So if we're interested in studying the large body phenotype, much like in the GWAS case, we really need more than one instance of it to have any statistical power. And thankfully, in the mammalian tree, we do have multiple instances of large body sizes evolving independently. And in that sense, the large body size is actually a convergent trait, which gives us a statistical power in order to understand the genotype-phenotype relationship. So in order to illustrate how we might use a convergent trait to do that, we can take this very kind of simple example of a convergent trait where we know what to expect in terms of what we might find biologically. So the trait we're looking at here is subterranean habitat, and it has evolved independently multiple times in the mammalian phylogeny, as illustrated here in this tree with the red branches. And you can see that visually, these species actually look very similar, having evolved a very similar body plan. And we can try to correlate this phenotype with the underlying sequences of these genomes in multiple different ways, but one very particularly effective way is to look at evolutionary rates. So in order to do this, we can take this protein-wide tree of evolutionary rates and contrast this with the same tree, but computed from a specific genetic element or a gene. So in this case, I'm showing the tree for LIM2, which is a lens intrinsic membrane protein. And as you can see, it looks very different for the protein-wide average, and it has actually this striking pattern of very long branches leading up to our subterranean species. And this, of course, makes sense because this gene is only needed to make the lens, so the structure in the eye. And one of the things all these species have in common is that they have highly degenerate eyes, as they don't need them to navigate their environment. We can visualize the differences between these two trees as the relative evolutionary rates plotted here. And in this view, we can see the top four most extreme changes in rate relative to the average are indeed our subterranean species. And if we look for this kind of pattern genome-wide, we can rank all of the genes and according to how much they correspond to this pattern. This rank list is highly, highly enriched for genes that have to do with diverse processes involved in eye formation and function. Now, one thing with this analysis is that while it's highly effective and can be applied to lots of different phenotypes, we did need to make a tree. And in order to make a tree, we needed to have an alignment. And alignment-based approaches definitely have some shortcomings if we're interested in analyzing non-coding regions. So we can think of kind of a non-coding element defined by either like as a FastCons region or based on functional genomics data, and this will typically be hundreds of base pairs long. But of course, the element itself is composed of multiple kind of basic units of regulation, which are TF binding motifs. And you can have multiple kinds of binding motifs and also repeat instances of the same motif, and the entire functional landscape of this is quite redundant, as it can be quite redundant as illustrated here. So you might actually have two motifs for the same transcription factor, and changing one of them while alters the sequence doesn't really alter the functional output of the sequence. And this results in this very interesting observation that over evolutionary time, what we can observe is what we call motif turnover. So this is quite well illustrated in this figure, where we're looking at the human and mouse genome at an orthologous and syntenic location, where in both cases we have a GATA1 binding event by ChIP-seq. And now if we zoom in here and we look at the actual sequence, and here's the alignment depicted for multiple species here, well, we can observe that while we are actually getting a reasonable alignment, the actual location of the GATA binding site has changed between the primate and the mouse lineage, with the functional output, especially from the GATA binding perspective, actually remaining the same. So at the sequence level, these sequences look different, but on the functional level, at least from the perspective of this one TF, they actually have not changed. So we've set out a method that takes all of this into account, and we call it alignment-free converge, or AFconverge, and it's a TF-centric approach to convergent analysis. So this method starts out with a set of regions for which we have orthologous sets across the genome. So they're not aligned, and they're not even necessarily alignable. But we have somehow determined that they're orthologous, usually by synteny. And we have a database of transcription factor PWMs. So using a database, we can score these regions, and we just record the maximum motif binding score, and with some filtering for no binding. And after we do this, this turns this data set into a region by motif by genome tensor. So at this point, we haven't used any phenotypic information. So given a specific phenotype and a phylogenetic tree, one of the really important steps of this analysis is we generate lots of null phenotypes which match this phenotype in a statistical way, but are not expected to produce any convergence signal. And we can, together with the null phenotypes, we can assess how well the values in this tensor are associated with the true phenotype of interest. So in the end, what we end up is a matrix that's a region by motif matrix, which now every point in here indicates a z-score for motif loss and gain associated with phenotype, calibrated relative to a bunch of null phenotypes that we have generated. And so you can think of kind of a specific example of this looking kind of like this, where if our phenotype of interest is body size, we're looking to a specific orthologous promoter. Here we have a motif that is on average gained as you increase in body size, and the red motif is on average lost, but we're not at all taking any position information into account, just the presence of absence of this motif signal. Now, in order to understand how well this method behaves, we actually go back to this the subterranean phenotype. It's a little bit different in this case because we've actually included the echolocating bats. Because from the perspective of vision loss, it's actually the same phenotype. Vision is not their, they have a reduced reliance on vision and very small and degenerate eyes. And so we're working with this large 241-way mammalian xenomid data set. and about 50,000 promoters, and we have a non-redundant TF cluster database of motifs. Looks like this, where redundant motifs are clustered into their consensus archetype. And so for this data set, we know what we expect. We expect that in these species that have vision loss, we expect degeneration in the protein coding sequence, degeneration or kind of relaxation of the evolutionary rate in the protein coding sequence of eye-related genes. And then we expect the same thing from promoters of eye-related genes. And so we can actually, even though we have quantified scores for a lot of different motifs, we can kind of quantify like a global motif loss score. And basically the way we do this is we compare a region of interest against all the other regions with a Wilcoxon rank sum test. And this basically gives us a ranking of promoters for overall loss and over like global loss and global gain. So we're analyzing the global loss, and we expect to see enrichment for vision-related functions. And so we assess this relative to cell type markers that mark various cells in important various cells in the eye. And this shows the enrichment for the enrichment that we observe. And so our method, AFconverge, is in blue. It's showing enrichment for most of these things and doing this usually quite a bit better than the competing methods. So AFconverge, which I talked about, which is alignment-based. AFforge is an alignment-free method, but it's supervised in the sense that you have to know which transcription factors you're looking for. And so for the most part, AFconverge performs the best in terms of finding eye-related elements. Okay, we can also, instead of looking kind of at the element or promoter view, we can do the same thing, but now we look at the motifs. So we can ask like which motifs have been globally lost or globally gained. Again, in this case, we're expecting loss, so that's the focus of the analysis. And we get a number of things that we expect. So we have a circadian pathway transcription factor, and also a master regulator of eye development. So now that we've kind of benchmarked the method, we can see that it generally works. We're going to look at a more interesting phenotype where we actually don't know what to expect. And the phenotype we're looking at is the body mass and longevity axis. So these two variables are highly correlated. This is lifespan. This is adult weight. So we actually take kind of an average approach. So we take this principal component. And the underlying alignment data set is the same as before. Okay, so on the left here, we're showing the kind of the p-value distribution that is calibrated against null phenotypes. And so from what we can observe, kind of the global statistical properties of the signal that we see, and we do see enrichment in both motif losses and motif gains. But there's actually more of an enrichment for small p-values for motif gains, and this was we were very happy to see that because large body size and long life are actually derived traits, so they do involve innovation relative to the ancestral trait. And just to give some examples of the outputs, so we can so this is a promoter for a specific gene, and in blue these are several significant motif gains associated with the evolution of the trait and some motif losses associated with the evolution of the trait. Okay, so we can do precisely the same kind of motif-centric analysis looking at global gains, which is what we have the most of. And there's quite a diverse number of motifs the diverse set of motifs that have been gained, but just to highlight a few, we see some early development patterning, Hawks, like in the Hawks family. We do see the pluripotency module, so the POU family and NANOG, some immunity-related things. So FOXO family, which have been implicated in longevity in a number of different organisms, and also a tumor suppressor SMAD4, which has been expanded in some longwing species. On the losses side, there's actually we have fewer losses overall and the fewer motifs, but there's a couple of interesting things to point out. So there's an overall loss of CG, just mononucleotide, and also CPG dinucleotide content. and there's a loss of a ubiquitous transcription activator YY1. Now, in order to kind of dive deeper into the matrix, the full region by motif matrix, we've decided to put it through a latent variable analysis, which kind of decomposes all of the diverse associations between motifs and regions that we have, and we use empirical based matrix factorization to do this. So after we do this, one thing that we notice immediately is that a lot of the effects is actually explained by the first two latent variables, which are interestingly highly anti-correlated with each other. And so, so we have latent variable 1 that corresponds to which is being driven by CG and CPG gains, and latent variable 2, which is driven by the same things, but instead these are losses. And so basically, we know that this pattern is lost kind of globally, but it's interesting to see there is actually a subset of genes which is it is gained. And there are there are other transcription factors on this axis, but what's interesting about these is that they kind of follow the CG pattern. So, for example, some like FOXN1 is very CG rich, and FOXC1 is very AT rich. One thing is that this kind of correlation between changes in CGs and CPGs has been shown before, and the correlation between those things and lifespan has been shown before, and there's multiple papers. You can actually make predictors based on this, predictors of mammalian lifespan. So this, in some sense, is a positive control. One thing is within the factor analysis model, we have a motif axis, and we also have a gene axis. So we can ask what genes are actually related to these latent variables. So if we focus on the loss, this is actually highly enriched for RNA processing and transcription. And on the gains, we actually have very few enrichments and just B cells and TNF. One thing that we thought was kind of interesting is this polymerase 3 is genes involved in polymerase 3 transcription initiation are experiencing CG and CPG loss. And actually, if you just Google this, one of the first things that comes up is this paper. If you Google RNA polymerase 3, one of the first things that comes up is this paper has been actually strongly implicated in controlling longevity. Just want to show one more interesting thing. We found this latent variable 8, which appears to be related to body size. It had literally one significant enrichment for a non-computational gene set, so regulation of BMP signaling. These are the transcription factors that were driving this latent variable. And the main one, this is the top score, is L-CoRL FOXR. And this is actually known to regulate body size, and it's in fact part of its NCBI gene summary. So just in summary, AFconverge is a TF-centric alignment-free method for evaluating convergent sequence change. AFconverge outperforms competing alignment-based and alignment-free methods when using a benchmarking phenotype with kind of known expectation, for which we use vision loss. And when we apply it to this phenotype that we don't understand well, the body size longevity axis. So our analysis implicates early development patterning, pluripotency regulation, transcription translation, and immunity as core mechanisms in experiencing convergent regulatory changes. And I would like to thank the funding sources, and thank you very much for listening.